Interesting discussion can be found in Science magazine, in department news, about a genomic study which raises concerns of others over race analysis. And here you probably recognize a UMAP plot, which we use many times in genomics for gene expression and other things. It is dated 23rd of February, so you can come and read it yourself. The whole title already explains the discussion that we deduce racism out of the UMAP. So the story is that people, quarter of a million US citizens, donated blood to some researchers, and then the researchers published a paper about genetic diversity of people across the country. It is necessary to properly treat patients. It's really not a nonsense research. But then there came an accusation that, first of all, there was a not probably representative selection of the US citizens, which is hard to tell, and usually ethnic minorities should be represented in a higher percentages in the population. Otherwise, the whole study will tell you nothing about minorities. So the bias is necessary for the study. For the rest, I can't say. And then the second accusation, which raised my eyebrows, was blaming the UMAP for the racism. Of course, indirectly. What is UMAP? I explain it more clearly in the script a bit later. But in words, it is a way of showing data in 2D, which are highly complex in their nature. So it is a simplification of the complexity of the data. There are many ways of simplification. The old style is PCA MDS, then somewhat later came tSNE, and the very latest achievement is a UMAP. So nothing is new, and scientists used it a lot. And now the claim is that UMAP is generating artifact which is actually do not exist. Then the same author says there is no easy way to represent this data is in 2D. Okay, everybody knows it. That's why UMAP is invented. Then another guy says, I am not a UMAP hater in all settings. Good luck. But I think it's misleading and potentially harmful for this specific problem. Really? So if the study was slightly different in different direction, then everybody would accept it. Is it what you say? I'm totally confused. Then the defendant says that the figure they presented is actually nothing new, and it was used many times by other respected agencies. It is so, and so on, so on. Let me show you the little script to illustrate the problem. Suppose we generate X series of data, 100 of lengths, 5 on average, 2.5 standard deviation, and then we generate another series called Y, also 100 with average 7.5 and standard deviation 2.5. Now we will split our output window in 4 parts to accommodate 4 plots. That's the meaning of this par MF row. And now we do the first plot, which is simply data X and Y as there are. Good. That's one series, and that's another. So I flip X and Y, and therefore I generate 2D data which are slightly different, but by and large they overlap in a lot. They are not separating well at all. To make it more believable by biologists, I will generate 10 times of gene expressions, or it can be some allele frequencies, and multiply these little differences 10 times in 10 different projections by these two loops, and then bind all the series together and then transpose, because this is how PCA likes to do. If you want to know how it looks, it's 10 genes and 50 columns. No, we have in total 50 dots of each color on the screen, and then we keep the same blue and red colors. So I call a library lemma because it is a very standard for gene expression. It has a PCA MDS conversion of this complex data. And now we do a standard classic transformation of this data into the MDS plot, which is nearly PCA. There are two versions of it, both give reasonably similar results. I mean PCA and MDS. So what happens now that because these little differences multiply 10 times, the collection of those differences between a series, blue and red series, is at the end increased, and so PCA plot, unlike to original data, is capable of separating blue and red clouds better. And if we go to a tSNE, which calculates based on PCA data, so we follow the classics, it is approximation, therefore it approximates to minimize the error, and then we plot it, and then you see that tSNE again is not much better in resolution, so this nasty 3 points still come in a wrong cloud. And now we go to the UMAP, and UMAP also approximates data out of PCA, and then we ask for it, and now we see that when differences between groups are small, then either PCA plot or tSNE or UMAP, they show pretty close differences. They are not very much different from one another. Attempt number 3. So you see that UMAP does not create something out of nothing. It is absurd. And at the end, a few more contrasting examples. Note that with increased distances between groups, UMAP will do better compression of those groups and higher distances between. That's what it is. So you see that this is probably the best out of all attempts, illustration that gradually, slowly, from original data to the PCA plot to the tSNE and UMAP, the distances between clouds are growing, the distances within clouds are shrinking, but it's not out of nothing, it's not out of the noise. At the end, some short statements. Before you do something big and public, always test your data for quality and filter all bad samples out of it. Secondly, UMAP should be used properly and carefully. UMAP is specifically made for very big data sets in order to make condensed clouds of data. That's the difference. There is always a tricky moment of clustering and classification of resulted clouds. we all know it. And only then you go public, and if somebody doesn't like data or UMAP, the person can choose not blaming data, blaming UMAP, or other way around. It's up to you, but it's not really a serious discussion anymore.